Thank you all for coming. As Nicole said, I'm really excited to tell you about some of the research we do in a couple buildings over from here. So in the Michael Smith Laboratories, and also in the Department of Biochemistry and Molecular Biology, which is a few more buildings over from that. So yeah, today I'll tell you what I think mussel glue is way cool. And so the reason why I think it's really cool is because I think we can use the chemistry that marine mussels use to glue themselves to things to treat blood vessels. And so here are some mussels glued onto this plant, and here's some blood vessels here. I don't think these ones are diseased, but there are lots of diseases of blood vessels, and so there's a connection here that I'll spend a few minutes of the talk telling you about. I think it may be a little bit of it may initially seem like a little bit of a stretch going from marine mussels to diseased blood vessels, but I'll tell you how this all works. So of course, marine mussels are way cool for other reasons. A lot of people think they taste great, but I'm not gonna, I hope you weren't expecting live demonstrations of this type of thing. I didn't bring any marine mussels to eat. I'm going to focus mostly today on the mussel chemistry and how they're related to adhesives and biological adhesives and blood vessels. So I'd like to start by telling you a couple problems. So a problem that marine mussels face in trying to survive, and then a problem that humans face in trying to survive for long periods of time. So a problem that mussels face is staying adhered to the ocean floor. So mussels want to be where the nutrients are and where other mussels are, and they don't swim in the ocean. They can't seek out other areas very easily. So what they want to do is they want to stay stuck to areas of nutrients and areas where their friends are. But this is a challenge, and I'll tell you how mussels have overcome this challenge. But it's related to this problem here that mussels face, and that's delivering drugs to diseased areas of our body. So disease is typically localized. I mean, most problems start in specific tissues or specific cells or specific organs, and so there's a huge enterprise trying to develop drugs to treat these diseases, of course. And one of the main challenges is how to get the drugs to the diseased area. So in pharmaceutical science and in biochemical research and biomedical research, people are developing all kinds of agents, small molecules and biomolecules and nucleic acids, all sorts of things that can bind and inhibit their biological targets and inhibit biological processes. But the main challenge between this, oftentimes this initial biochemical research and actually getting a drug to the clinic is getting the drug to the disease region of the body. So this is a problem that we face in treating disease. And so as I've been saying, there's a connection here, and I'll tell you more about what it is. Okay, so a little bit about the marine mussel problem. As I mentioned, some marine mussels, they want to stay where the nutrients are and where their friends are. But this isn't, it doesn't seem like it'd be that hard to do in situations like this, where the ocean where it's a nice calm day and, there's no waves and it's nice weather. I mean, the mussels can stay here. They can stay stuck on on these pipes or this dock. I'm not sure what it is. Where the mussels can stay stuck to different surfaces in the ocean. But what happens? What happens when this happens? When there's huge waves coming and crashing down on these mussels. I mean, how do they stay there? And they do. We know that they do. We know these mussels are hard to collect. If you ever collected these marine mussels before, I mean, they're a little bit. I mean, you can rip them off the surface, and people do off of rocks and plants, but they definitely stay stuck pretty well. So I'll tell you, there's been a lot of research about how these mussels stayed here, and I'll tell you a little bit about how these researchers what they found is the answer to this. But jump back to this problem that the humans face. That's delivering drugs to diseased regions of the body. So here's a couple examples of diseased blood vessels. And, people think that if we could get drugs to these particular areas of vessels, then we can treat them. So I'll go through these. One of them is atherosclerotic plaques. And so what we're looking at here is this red part. This is an outline of a blood vessel. If we look at the whole blood vessel, it'd be really large. So this is just a small, tiny little microscope image of this blood vessel. And there's an atherosclerotic plaque in here. So there's a, so there's a plaque that's coated this blood vessel, and this plaque, it can become a big problem. So if this plaque, if it ruptures or bursts, it can cause the blood that would be here. It would cause this blood to clot, and if it clots, this can lead to a heart attack or stroke. So a big problem. Another area of diseased blood vessels is an aneurysm. So this is where a blood vessel has become weakened and it can balloon out, and this blood vessel can burst, and that's a huge problem. So tumors, right, are a big problem. So this is local area disease. Tumors usually start in one area, and if they become cancerous and if they spread throughout the body, it's a huge issue. But initially they're small, and they start off just in one little spot. And here's another area disease, arteriovenous malformations. These are jumbles of arteries and veins. And so there's lots of therapies that people think could treat these, but the problem is, how do we get them to this area of the body? And this is a huge question in biomedical research. So how do we target these areas? If we come up with a drug that can treat this plaque, how do we get it to this 1 millimeter section of the blood vessel when there's actually kilometers of vessels? How do we get it right there? And people have been developing all kinds of technologies to do this, but it's still a huge problem. So I don't know if you have any ideas. I'd like to hear them. But what do you guys think? I mean, how can we how do we get these drugs to go to these areas of disease? I mean, there's no there's no like right answer or wrong answer, but yeah.
Maybe you could build a tiny robot that could tell where that is, and it could go through maybe like your mouth or something and find it. Like a really, really tiny robot.
A really tiny one? Yeah, I don't know. I mean, I think it's pretty reasonable. So, have you heard of platelets before? Yeah. So other cells kind of do this. Other cells are like little robots. I mean, they're not made of metal, but yeah, there's all kind this is what lots of cells do, right? Cells go through our blood vessels and search out. It's like macrophages or other types of cells like platelets, and yeah, so they go out and they search for areas of disease, and when they get there, they dump out their contents. They dump out contents that can help heal that area. So yeah, so that's so people would like to develop systems like this that can biological robots that can seek out disease. So that's yeah, that's a good idea. Are there any others? Pardon me? Oh, ultrasonic waves? Yeah. Ultrasound. Ultrasonic waves are they're used in drug delivery. So so lots of ultrasound imaging is done with these like micro bubbles that are injected into the bloodstream, and so people found that at the right ultrasonic frequency that you can burst these bubbles, and so people have tried to put drugs on the outside of these gaseous bubbles and deliver them, and use ultrasound to locally go in, right? So you direct the ultrasound waves right to this area where there's a tumor. You try to burst these bubbles and deliver drugs to the tumor. So that's it's definitely an area of big exploration at this point. And so ultrasound is used for imaging. I don't think there's any clinical uses yet for drug delivery, but people are working hard towards it. So yeah, definitely. Any others you guys know about or think may work? Pardon? Infusions. Yeah. So yeah, direct infusion. You can go in a lot of these areas. You can go in with tubing with catheters. So you can take these long tubing and feed them through arteries. And clinicians, over in the UBC hospital do this now. So they feed these catheters through blood vessels, and it's amazing how they can wind them all the way through and go to these areas of disease and locally inject the agents. So you can do this, and that's pretty close to what to the subject and tell you about today. Because we use these catheters to go in and locally deposit agents. And that works, but it only works for some agents. So it works for agents that are fast acting and agents that you only really need to do this every once in a while. So a once a month kind of dose or a one-time dose. Now if you need your drug to be continuously there at the site, then it's more difficult to do it with a catheter, right? You can't go in. You can't can't give someone a catheterization 3 times a day like you can take a pill 3 times a day. So what I'd really like to do is go with a catheter and deposit something that can stay put, right? That can stay in this area. So we go in, we put the drug here, and the drug stays locally, stays put at this area. And so that's there's the question that you guys had some great answers to. And so that's a potential solution, is to deposit a drug-eluting material onto the blood vessels using a catheter-based approach. And so, yeah, with some drugs, you can go in the catheter, locally deposit them, in this tumor, but they just flow away. There's blood flow, just washes away. But if we could put a material there that slowly released the drug out of there, then it could potentially treat these areas. And so, of course, the challenge here is, how do we keep these materials stuck in place? Right? So if the blood is rushing through there, there's a really high force, the blood's there's pumping through there, pushing it through there. How do we keep it in place? And so there's an opportunity here, that comes from mussels. So mussels, right? They do this. They stay put in place, and so let's figure out how they do it. Let's figure out how they stick to these surfaces, and maybe we can stick materials to the inside of blood vessels. So that's the idea. So I don't know if it seems like much of a stretch. We're talking about mussels sticking and drugs sticking to blood vessels. So, but I'm not sure. So maybe you're with me, but I just want to make sure. So are there any similarities between these 2 environments? So the ocean and our vasculature. So is it clear what the similarities are here? What's that? Fluid, right? Yeah, they're underwater. Yep, and yep, they're underwater, and there's strong flow, right? So these mussels, right? They're in the ocean. It's underwater. So the glues that they use work well underwater, and the glues that we would need to stick things to blood vessels, they have to work well underwater. And then there's strong flow, right? So the mussels, waves can crash over them. The mussels stay stuck. So it's underwater glue, and it's really, really strong. And so, so this is why we think there may be an opportunity. If we understand what mussels do, we learn how they do it, and maybe we can use the same technology to glue things to blood vessels, or other areas. There's lots of applications for biological glues, and so we think if we can develop something that sticks to blood vessels, excuse me, should stick to other parts. So, so just a little bit about mussel adhesion. There's a lot known about mussel adhesion. And so people, for the last 15 years or so, people have been really interested in how mussels do it, and people have learned a lot. So what's known is that it's a very versatile adhesion. So these mussels can actually stick to all sorts of different things, and it's water resistant, or it works well underwater. And so some of the natural things that mussels stick to, so they stick to rocks. This is probably what you most often will find them stuck to if you're out collecting them in the ocean. But they also stick to all kinds of natural organic materials. So they stick to plants well. They can stick to feathers and fish skin. So in their natural setting, they stick to all sorts of stuff. In their natural setting, they also stick to some man-made things like pipes. So they can stick to all kinds of different metals, and they can stick to like titanium and these things used in electronics industry, and they can stick to semiconductors, and they can stick to synthetic organic materials too. So like shown here in this example, these mussels stuck to this tire, this plastic tire, right? So they can stick to all kinds of different things, even to Teflon. So polytetrafluoroethylene is a type of Teflon. So Teflon's a really inert material, and people use it because nothing sticks to it. Well, these mussels can stick to it. So it's this is a pretty amazing glue. It's really adhesive, and it works underwater. So how do they do it? What does this glue look like, and how do they apply it? So how do mussels apply the glue? And so here's some images of the ways that we often apply glues in our home and around for different applications. Mussels do it a little bit differently. So mussels, we look at the anatomy of the mussels. They have these feet, right? They actually have, each mussel has one of these things, these feet. And what they do is they stick these feet out onto a surface. So here's the surface. So they stick these feet out. And then what they do is they they fill this this foot is hollow. At least it has a thin tube in it. So they fill this foot with a glue material. And so here's what one of these glue materials look like. It's this Bissell thread, this strand. This is what is currently inside of this foot. So the muscle comes down, it puts its foot on the thing, fills it up with this material, and then it retracts the foot. And it does it over and over and over again. And so the foot stuck this one down, now it's sticking another one down, and then it'll keep going, and it'll stick down a bunch of these, maybe 5 or 10 or 20 or 30 of these. But and this is what this Bissell is what actually keeps the muscle stuck to the surface. It's just this thin tissue with this little more adhesive part on the bottom. And so this is how they applied the glue. But so there's just a little bit more about the anatomy that these muscles have. So here's the foot. So this is a foot that's been retracted. So it's now inside of this. Here's the muscle. Here's the muscle here. Here's like a rock or some other surface that it's stuck to. Yeah. So there's these Bissell threads, and then there's this area here, this plaque, where it's really making contact with the surface. And yeah, so you can see the muscle makes a bunch of these. So it'll go down and just keep sticking and sticking. And if some of them break, the muscle can go and stick some more down. But and so if you're ever collecting mussels, you'd be breaking these. You're trying to break these threads. Sometimes it could be challenging. You have to twist and turn, and it can be a little tricky. So what's actually in these Bissells? What is the what's the chemistry? What's the biochemistry of these things? If we're going to try to mimic these this material, we need to know what's in it, and where does this adhesion come from? So there's man-made glues that are adhesive underwater. So super glue, they're all pretty familiar with. So that works well in an aqueous environment. But these are based on small molecules and really reactive chemistry, not something that a biological organism would use, or the biological system like us could take advantage of. So what's in this? What's the magic that the mussels use to do it? And so people have taken these things and broken them apart. They've purified all the proteins and analyzed all the proteins and looked and see looked and found what the composition of these structures are. And so what they find is that there's some threads, some collagen threads in here. But what they find is particularly in this the plaque, this part that sticks to the surface, there's a bunch of proteins in here that are all really interesting. And they're here they're MFPs, so it stands for muscle foot protein. And so there's MFP1 and 2 and 3 and 4 and 5 and 6. There's probably others too. And so in this structure here, it's full of these proteins, right? These muscle foot proteins. And people have looked at these and tried to understand, what's special about these proteins. So people found that it's actually the protein that's the sticky part. But what is it about the protein that's the sticky part? And so here's so the proteins. So human proteins have 20 amino acids, right? And so if people went and they looked and tried to characterize what amino acids are in these proteins, what they found is that there's some amino acids in here that humans don't have in their proteins, and it shows up all over the place. And so this amino acid here in red is something that doesn't show up in our proteins. And so it looks kind of similar to an amino acid we have. So we have a amino acid, tyrosine, that has the same thing, except it's missing this hydroxy group. But these muscle proteins, particularly in their feet, they have this extra hydroxy group, and they have a ton of these amino acids that we don't. So they're just like all over the place in the muscle. And so what this amino acid is dihydroxyphenylalanine. It's called DOPA. It's kind of interesting. DOPA shows up. Humans have DOPA, so humans have this amino acid. It just doesn't get incorporated into our proteins. So DOPA is in our brains. It's a neurotransmitter, but we haven't found it in any human proteins yet. I mean, I wouldn't be surprised if we find it, but no one's found it yet. But so this amino acid just all over in these proteins. So what is it doing? Do we know anything about this unnatural amino acid, or this amino acid doesn't occur in humans? What do we know about the chemistry and what it does? And so we do know a little bit about it, and actually it comes from tree science, because DOPA is a type of catecholamine. So this here's DOPA here, and so this group here is a is called a the chemical name for this moiety is a catechol, and it has an amine, and so it's a catecholamine. And these things show up in trees all the time. It's actually the glue that trees use to keep all their cells, keep everything stuck together. And so tannins that trees use as an adhesive, and the same tannins that show up in red wine that are antioxidants. So they're full of these catecholamines. And so people know quite a bit about these catechols and the why they're adhesive. And so these catechols, they can stick to metals really well. So they can these two hydroxy groups, they can chelate to rocks and other metals. They stick to metals. But also if they get oxidized, then it forms this other chemical group that's really reactive. So this chemical group can bind to other surfaces containing like nitrogens and surfaces containing sulfurs. So it can bind there. It can bind irreversibly. So this is a covalent bond that doesn't break. So these catechols can just really stick really well to surfaces. And so that's what the mussel is doing. So the mussel, it incorporates these tree, the mussel, mimics the tree chemistry in order to adhere really well to the rocks. So, in fact, when people started learning the mussels have this, people got really excited again about catecholamines and started wondering, well, can we take these catecholamines and coat them on other things? And so a friend of mine, Haitian Lee, what he found is he could take DOPA and other catecholamines, not the muscle proteins and not the muscle proteins, but just a small molecule. He could take this small molecule that mussels that are in muscle proteins, and he could stick it in all kinds of surfaces. And this DOPA, when it sticks, it turns brown and red. And so when he took it and he plated this piece of titanium that used to be gray, all of a sudden it turned red because it was coated with this chemical that mussels have. And if he took a dime and dipped it in this solution, the dime turned looked more like a penny, but it's still a dime. It just has this red-brown color on it. And he could do this with all sorts of different materials and plastics and all sorts of things. So so DOPA sticks to a variety of surfaces. So so the big question that I want to ask is, so can mussel-inspired materials, can they stick to blood vessels? Right. So can we take materials that look like muscle proteins and stick them to these blood vessel walls that we want to? And so I can tell you how we did it, but just to give you the answer. So the answer is yes. And so we made these materials that looked like marine muscle proteins in some ways. So the proteins themselves wouldn't work very well in biological applications. A lot of times when you take proteins from other organisms and put it inside our body, it gets rejected, right? So our immune cells and our inflammatory cells, they attack it, they clear it out. It doesn't work well. And so instead of taking the actual muscle protein, what we did is we took these inert polymers. And so this is a hyaluronic acid. This is a polymer that's already in our bodies at high concentrations. And there's another one, alginate. It's a really inert material that's found in algae. So we took these inert materials, and then we changed them to look more like the marine mussels. And so we took these catecholamines, these DOPA-like molecules, and attached them to these polymers. So we took these inert polymers, and then we switched them to make them look a little bit more like these mussel adhesives. And so when we took these materials and tried to coat them on blood vessels, what we found is that, they would stay stuck to the blood vessel wall. And so here's a couple images of that. Here are two carotid arteries attached to the heart. And so if we go in higher magnification, we can see these again here, these two carotid arteries. And this one here we've coated with this muscle mimicking gel. And so what we did is we took these gels and we also blended some quantum dots. These are some really tiny nanometer size particles that fluoresce really strongly. And so we took these particles, put them into this muscle mimicking gel, and then coated them on the blood vessel wall. And so wherever we saw this red fluorescence, and that's where these materials were. And so we can see as we zoom in here. Here's a blood vessel that was coated. Here's a blood vessel that wasn't coated, and so you can see that there's this material is stuck on it. There's this red fluorescence coming from this blood vessel, and so so we've adhered this muscle mimicking material to the blood vessel wall. So Yeah. So why? So I think it's really cool that we could we could mimic the marine muscle chemistry and put it onto blood vessels. But is it useful for anything? So I just want to come back to this part about this point about treating. See if I can lower this down. Yeah. Treating diseased blood vessels, and particularly treating atherosclerotic plaques. Okay, and so the idea is that so we can adhere this material to blood vessels. Can we adhere them to diseased areas? And so this is what I wanted to test. And I wanted to look at atherosclerotic plaques and vessels that had atherosclerotic plaques. And the reason for that is because atherosclerosis and these plaques is just a huge, huge problem. So about 70% of the mortality from coronary artery disease results from plaque rupture. So all the heart attacks and strokes that you hear about, almost all of them are caused by the critical event that leads to a heart attack and stroke is the rupture of atherosclerotic plaques. So when these plaques rupture, then it'll clot, the blood will clot, and the clot will break off and then occlude downstream vessels. So it's a huge problem. There's a particular type of plaque that we're interested in. So just to kind of show you what we're looking at here. So here are two different blood vessels. And here's the blood vessel. This is kind of so the blood vessel is really long, and we took like a section through it. Well, this is someone else's section. So someone took a section through it and stained it, and then they could see what kind of cells are present. So here's the blood vessel wall, the outline here in purple. And here is a big old gray, brown, crusty old plaque that's stuck in there. Now, this plaque here, it gets a lot of press, and that's because this plaque here can get treated by a stent. So a stent is a device that goes in and opens up the blood vessel wall. But stents cause a lot of damage, right? And they have these metal struts that come and push open the blood vessel wall. So we wanted to come with, we thought that this mussel adhesive became a much more benign way to treat plaques, particularly plaques like this that are more susceptible to rupture. These big plaques, although they cause medical issues such as angina, they actually don't. They're not the causes of heart attacks and strokes. It's actually these small plaques that are the culprits. These ones, they're not necessarily small, but they can be small. They're the ones that are really soft. So they're really soft and they're prone to rupture. And so if this plaque here, if it breaks apart, then like as I said, the blood will clot. This can cause a heart attack and stroke. So the idea was, well, what do we coat over this plaque? So we're going to take these muscle mimicking gels, we're going to coat it over this region, and deliver drugs from the gel into the atherosclerotic plaque. And so that's what we set out to do. So we want to coat over these plaques and deliver drugs into these plaques to heal them. Okay. And so just once again, what the idea is, the way that we make these materials is we take a really inert material and we mimic the marine mussels by incorporating this DOPA-like chemistry. So we attach that to this. This is the schematic of the polymer. We attach the DOPA to it. Then what we do is we take this polymer and we cross-link it right onto the blood vessel wall. And in this gel, we have particles that can release therapeutics out of the gel into the blood vessel. So that's what that was the idea. The first question is, well, do these materials, do they really stick well under blood flow, right? And we know the muscle does it, and the hypothesis was that these muscle mimicking gels would stick. But we wanted to test that. We wanted to test if the gels would stick. We wanted to quantify, how well do they stick. And so to do that, we did a kind of a simple, more simple experiment, in that we made a device that looked like this. So it's just a plastic chip, and it has this channel running down through it. And in this channel, we coated it with endothelial cells. These are the cells that line blood vessels. And so what we're doing here is we're mimicking blood vessels and mimicking the surface of blood vessels on this chip. And the reason we do this is because we can really carefully control the blood flow through it. So we can really carefully quantify how adhesive the material is. And so we make these biomimetic blood vessels. Then we take our biomimetic gel and we inject it in there. So we put a catheter into this plastic device. We inject some gel out of there, allow the gel to stick to the blood vessel wall, or excuse me, this channel wall. Then remove the catheter, leaving a sheath of the gel around the endothelial cells. Then what we do is we start flowing blood through here at different flow rates, and we look and see how much gel is attached at different flow rates. What we see is when we take this non-muscle mimicking gel, when we put it in here, here is these green and purple particles coated on the channel. What we see is when we start turning up the flow rate, the gel breaks off. So that this gel, it doesn't look like mussels. It breaks right off. However, this alginate catechol, this is the one that does look like mussels. It stays stuck there. And so when we start turning up the flow rate, it stays stuck. And so when we get to flow rates that are similar to physiological flow rates, it's still there. It's not breaking off. It's still stuck. And even when we turn it up even higher, it's staying put. The gel doesn't break off. And so we thought this was great. The material seems to adhere under flow at flow rates that are similar to what we see in our blood vessels. So we wanted to really test, how adhesive it was. And so the way that people usually measure adhesiveness is they take 2 plates. Here's 2 plates here. So they take them and they kind of glue them together. Then they pull them apart, and they use an instrument that measures the amount of force that it takes to pull the plates apart. So they pull these apart and measure how much force it takes. And so what we find is when we use this adhesive gel, that it takes a lot of force to pull them apart, at least a lot more force than what's experienced in our blood vessels. And so the force that it takes is the shear stress. It's about 1,000 times more than the force that the blood exerts on the blood vessel. And so the material is really adhesive on the scale of forces that are seen inside of blood vessels. And so, the way that we actually get these things into the blood vessels, as I mentioned, is a catheter. So these are these plastic tubes. Here are these are different than the tubes that are used in the clinic. That they use in the EBC clinic. These are much smaller. But we use these plastic tubes that have holes in the side. So we put them into the blood vessel, and we inject the gel out of the holes onto the blood vessel wall. They remove the catheter, leaving the gel there. Okay. And so do these things, do they stay stuck under blood flow? And do they, how well do they adhere? And do they adhere for a long period of time? This is really the big question. I mean, if the material just broke off, that would be a huge problem, right? If you have this gel stuck on a blood vessel wall, and if after a day or so, if the gel broke off, it would go downstream and block other blood vessels, right? It would embolize tissue beneath there. That would be a huge issue. So it was really critical to make sure these gels didn't break off. And they luckily, they don't. So these are really, really adhesive. It's a surprise that marine mussels can stick under the ocean. It was it was a big surprise that these gels could adhere to blood vessels. So here's some images, some microscopy images of that. So here's a blood vessel here. In red, there's blood flowing through here, and in green is this gel. So the gel is adhered there while there's blood flowing over it. We can cut this blood. We can take the blood vessel out and freeze it and cut it into sections. And what we can see is so here's a section of a blood vessel. Here's the blood vessel wall here. Here's the lumen. Here's where blood would flow through. And then here's the gel. So it's just a nice thin coating of the gel on the blood vessel wall. In some cases, you can see here it looks a little bit patchy. Part of that is the imaging techniques, aren't perfect. But another part of that is, it's not a completely uniform. But we were surprised how uniform the gel could be right along the blood vessel wall. And the gel stays there, right? So it was a big question. Does the gel break off? Does it change shape? Does the tissue wrap around it and do weird things to it? But it seems pretty stable. So here's the gel. It was coated on one day. It came back, on day 5 and looked, and what we saw was that the gel is still there. And remarkably, it still had the same shape. So the gel's still there. It's not breaking off. It's not clumping up. It's not getting the tissue isn't doing anything really. The blood vessel isn't doing anything really strange to it. It's still there. The material actually stays stuck for 30 days. So in this graph here, what we're looking at is the amount of gel, and then we're looking at different time points. And so at day 1, the amount of gel is here. And so we look at day 5 and day 30. There's still the same amount of gel there. So this is in a large group of blood vessels, and so the gel is not going anywhere, even for 4 months. So we have the gel there. We come back 4 months later and look. It's still there. It's not going anywhere. And so how does it stay stuck? And so there's actually other cells that grow over it. And so what we can, what we can do is we can take the blood vessels out and really, carefully we can take pieces of blood vessels and stain them. So we can add different dyes to them and see what color cells are there, and that can tell us what type of cells are there. And so let us know, are these blood vessel cells? Are they platelets? Are they macrophages? Are they other immune cells? So we can go and look. And so what we see is that this gel that's right here, it gets coated with a healthy layer of cells. So endothelial cells and smooth muscle cells get coated over it. And luckily, inflammatory cells aren't there. So there's not, this gel isn't getting rejected by the blood vessel. So there's not all kinds of inflammation, and it's not being attacked by the inflammatory system and the immune system. But the gel is still there, and there seems to be a healthy biological response to it. So this is something we were really surprised to see. And so now I want to see, okay, so we have this gel, it can stay stuck, and can we release things out of it? Can we put small molecules in here? Can we put biological molecules in here? Can we release drugs out of this gel? And so we can use more microscopy to look at this. What we do though is we can take a molecule that fluoresces and put it inside of particles that slowly degrade and put these in the gel. So here's the gel here in. There's purple particles that help us see the gel. Here's the blood vessel. Here's the blood vessel wall. Here's the lumen. Here's kind of the hole in the blood vessel where blood flows through. And so here's these purple particles are incorporated into the gel. There's also these green particles. Now these green particles are another type of they're a type of it's a particular type of plastic that degrades over time. So when it's in water, it slowly degrades. And the green that you're seeing is a small molecule. It's kind of similar to a small molecule drug. And so, as what we see is that we can coat these drug-eluting particles on here, and over time, the small molecules they come out of these particles and they go into the blood vessel. And so this red is the blood vessel, and so you can see that there's some green in the blood vessel wall that's come out of these particles and gone into the blood vessel. So we can adhere materials; they stay stuck forever. You can release small molecules out of these, and can we treat atherosclerotic plaques? Is the big question we're trying to answer. First part of that is, can we actually coat plaques? Do plaques look like other blood vessels? They look different, but can we actually coat them? And so we can. So what we're looking at here is another microscopy image of a blood vessel. Here's the blood vessel here, and this blood vessel has an atherosclerotic plaque right here. And we know that because we can inject dyes into the system that sense plaque. So when they're in the presence of a plaque, they start lighting up, right? They actually get cleaved by enzymes in the plaque, and so they light up. So it turns blue, and so here we know that there's the plaque right here. Then we can go in and we can coat the gel on it, and so that's here. The yellow is the gel. This gel actually coats the entire plaque. We only see it where the plaque is thinner, but we can actually see the gel over the entire part. And so just one more slide on kind of the answer. This is the last data slide, but there's a ton of there's a ton of data. I'm not going to go into the details here, but the main point I want to get at is that we can reduce inflammation in the plaque, right? So it's all about when you're treating plaques, it's all vulnerable plaques like this that are at risk of rupturing. It's all about reducing the inflammation there and increasing the thickness of smooth muscle cells on the wall. And so this is a bunch of data and just like quantifying that we could do that. So I'm not going to go through all of it, but what we see is that the smooth muscle cells, the cap over the plaque has gotten thicker. So in the treated blood vessels, there's a thicker plaque of these smooth muscle cells protecting it, making a thicker cap so that the plaque isn't as susceptible to rupture. And there's less inflammation in the plaque. So here we're measuring the mRNA in the plaque and measuring these cytokines, these other components that are all markers of inflammation. And so what we see is that there's less of these inflammatory molecules present in the plaque. And when we cut these vessels up, do histology, use microscopy, and look at the blood vessels, we see that these plaques. Here's a plaque here, and here's there's a nice thicker layer of the smooth muscle cells over it. And there's fewer inflammatory cells in the plaque. There's a healthy endothelial layer, and there's less this other marker of inflammation. So it's not you don't have to follow along with all the data here, but the point is that there's less inflammation there, and there's a thicker fibrous cap over the plaque. And so the plaque is less vulnerable to rupture at this point. And so that's the last data slide. So just have a couple conclusions here. And so one is that mussel-inspired adhesives can stick to a variety of surfaces. Right? So mussels stick to a variety of surfaces. The small molecules that are part of the proteins these mussels are really adhesive, and polymers that look like muscle proteins in some ways are also really sticky, and they can even stick to the inside of blood vessels under blood flow, so they can stick really well. The conclusion is that adhesive gels can stick and heal plaques, but by shielding them and releasing drugs into them, so we can actually take the chemistry that marine mussels use, put them into these synthetic polymers, and then we can coat blood vessels with them and heal, heal atherosclerotic plaques. And so I hope this is the conclusion that I've drawn. I hope you can in some way draw this too. Is that mussels and their glues are way cool. So just want to acknowledge a lot of the. I told you a lot of work that was going on here, and most of it I didn't do myself, and so. A lot of the work in the second part, I did do, and so that was when I was a postdoctoral fellow in Dan Anderson's and Robert Langer's labs. This was at MIT, and we had a lot of collaborators there, and still have collaborators. And a lot of the images and the background data from the beginning, about mussels come from these researchers. So Haitian Lee and Bruce Lee and Philip Messersmith and Herbert Waite, and many other images I took from the internet, and I'm not sure who the artists were, but. I'd like to give them credit. And here are the members of my lab who did a lot of the work that I'm showing. So I'm really glad that you came and let me share this data with you, and happy to take questions if you have any others. So, okay, all right. Thanks again for coming. Thank you.